morning, everyone. On behalf of the editorial office of Myeloid Cells, welcome to today's webinar. We are delighted to have you all here. To ensure a smooth session, we kindly ask everyone to keep your microphone muted during the presentation. Today's session will begin with a talk from Professor Julia Szyszkowska, followed by a discussion part hosted by Dr. Camille Belleriot and Professor Jean-Louis Mege. First, please allow me to make a short introduction. Myeloid Cells is led by our two editors-in-chief, Professor Florence Jean Hawks and Professor Laiguan Huang, who are internationally recognized experts in myeloid cells biology. Our editorial board currently includes 40 leading scientists from 13 countries, representing top research institutions worldwide, including National University of Singapore, Yale University School of Medicine, National Institutes of Health, and Chinese Academy of Science, ensuring a strong academic leadership for the journal. And today's lecture is in the topic of metabolic programming of macrophages. We are honored to introduce our speaker for today's lecture, Professor Julia Szyszkowska. She's from University of Heidelberg. Her research focuses on macrophage biology, particularly their roles in cancer, chronic inflammation, and metabolic diseases, with an emphasis on translation strategies for macrophage reprogramming. And we are honored to have the discussion guests, Dr. Camille Belleriot and Professor Jean-Louis Mege, to join us for today's webinar. Professor Jean-Louis Mege is a professor at Aix-Marseille University, specializing in immunology and infectious disease, with extensive experience in research, clinical practice, and training in human pathology. His work focuses on macrophage biology, host pathogen interactions, and immune modulation, with nearly 250 publications on topics, including intracellular pathogens, macrophage polarizations, and immune biomarkers. And also, Dr. Camille Belleriot is a research scientist at Gustave Roussy, specializing in oncoimmunology, with a focus on how metabolic reprogramming of macrophages influences tumor progression, particularly in obesity-associated cancers, using single-cell transcriptomics and integrative computational approaches. It is now to welcome our speaker, Professor Julia Szyszkowska. Professor Szyszkowska, the floor is yours. You can just share your slides.
Thank you very much for organization with great meetings and also for inviting excellent experts in the field to discuss my presentation at the end. So I would like also to thank you for organizing the first lecture, which we had a month ago, because it was very practically oriented and with clear solution and clear translational task. So I think my lecture will be different because I will rather share work in progress, no ready solutions, but I would like to open some points for discussion. And this macrophage metabolism will be like a last part of my talk with some unpublished data, which I would like to share with potential cooperation partners also, because I believe we need to join forces and to understand how this basic knowledge can be applied. So my deep background is macrophage polarization. I'm studying this since 2001 already. And I would like to start the lecture that macrophages in tissues have embryonic origin, so they come from yolk sac. The most the primary cells which migrate, myeloid precursors which migrate to brain, build microglia. Others go first to liver and make fetal monocytes, which are distributed through the bloodstream in different organs. And you can already think about that even embryonic metabolism of mother and embryon can potentially can already affect the immunity in our tissues if there is a metabolic memory. And then during the in the adult body, we have constant influx of monocytes which come from bone marrow, and they go to circulation. And if these monocytes migrate to tissues, they will reconstitute resident tissue macrophages. So this is probably a slow-running process. But if we have infection or tumor, trauma, myocardial infarctions, monocytes will be very actively recruited. So this means if we have some metabolic programming in these precursors, or if monocytes see some metabolic parameter factors in blood circulation, they can also acquire the program which will affect how they will perform their primary functions, defense and healing. So, and macrophages in tissue are very active, resident tissue macrophages, in terms of maintaining of homeostasis. They sense the danger in trauma or pathogen directly in the tissue. They start an acute inflammatory reaction, which can involve or not involve adaptive immunity, but they will orchestrate influx of next monocytes, also neutrophils here. And then they sense, actually, they have ability to sense the point that resolution of inflammation should start and healing should start, and then to restore tissue homeostasis. But if this process from acute inflammation to resolution of inflammation is not efficiently progressing, then we can have a state of chronic inflammation, which is kind of oscillation between inflammation and healing. And the big question is how this chronic inflammation is linked to the detrimental metabolic processes in macrophages, because this chronic inflammation will make a basis for cardiometabolic pathology, for cancer, also for neurodegeneration. Today, I will make as an example the history of therapeutic approaches developed for cancer, because in cancer, in tumors, tumor-associated macrophages are the best investigated in terms how to target them, which approaches is possible to do to instruct them to do the right job. So, and this is just a demonstration why macrophages in tumors are important, because they constitute in each type of tumor, they are infiltrating, there's almost not impossible to imagine solid tumor where macrophages are not infiltrating. They build their subpopulations from the resident macrophages and from incoming monocytes. And in majority of tumors, they correlate with bad prognosis, amount of macrophages, and also and then M2 skewed phenotype. However, there's one cancer, and this colorectal cancer, where we have much more complex situation. So we summarized in our recent review, we summarized what we know about macrophage biomarkers and subpopulations in such a way that we used only human data here and made analysis of subpopulations which correlates with unfavorable cancer prognosis, correlations with favorable cancer prognosis, with inhibition of anti-cancer therapy efficiency, and also cooperating with anti-cancer therapy. And this is very interesting because if you go deeply to the cohort studies, I would say unexpected, uncommon messages can come. So correlation with favorable cancer prognosis in total amount of macrophages is it was found by a number of studies for colorectal cancer. So this means in colorectal cancer, majority of macrophages keep the possibility to fight against cancer. And also some subpopulations, for example, for LER2, plus for breast cancer, TREM2 for head and neck cancer, and HIT1 for head and neck cancer, they also correlate with favorable prognosis. And of course, it raises the question, why these macrophages probably keep the ability to fight against cancer. And also, if you go for cooperation with anti-cancer therapy, there are studies which indicate that some macrophages can cooperate, but these are more like isolated reports. And of course, as you see, and this is a common knowledge, accepted knowledge, that majority of subpopulations cooperate with unfavorable prognosis, so correlate with unfavorable prognosis, and also inhibit different therapeutic approaches, including chemotherapy and more advanced immunotherapy. So our studies, a little bit of history of our lab. So our studies were focused on scavenger receptors for a long time, and this is why we've primarily investigated biomarkers which indicate for pro-tumor or anti-tumor activity of macrophages. So, and scavenger receptors here are very good biomarkers because they are transmembrane proteins, and they are very stably expressed. So this means it's easy to stain by the simplest immunohistology in paraffin tissues. However, if you look for the functions in tumors, these functions are not, I would say, simply to explain, because scavenger receptors can internalize something, including some lipid components. They can sort these internalized particles in different compartments, and there are scavenger receptors, and this was my work for a long time, like stabilin-1, which can also sort newly synthesized protein to bring them outside of macrophage. So this means even if we take scavenger receptors indicators, this does not mean that their function is pro or anti-tumoral. Sometimes it's just a manifestation, but when you think about targeting scavenging receptors function, I would say, despite that they're very important for metabolism, I would not go in this way because they are redundant in their ligand repertoire. They have the buffer function of each other, and they are, from my point of view, not decision making, but like optimizing the environment. However, they are very good if you would like to bring some therapeutic particles, for example, to a very specific macrophage subpopulation, because you can take 2 or 3 biomarkers of bad macrophage subpopulation, and these scavenger receptors will be actually the determinants for targeting. And if we look for what tumor-associated macrophages do in cancer, so they start from the beginning until the last metastatic stage. They have a lot of pro-cancer functions. These functions are extremely, I would say, complex and have a lot of mediators. For example, for initiation, if macrophages are instead of chronic inflammation and produce reactive oxygen species, so they create optimal environment for mutagenesis, and some transformed cells escape local immune control, can start to proliferate. And then they produce multiple growth factors and cytokines, which will support just primary proliferation of cancer cells. And when it comes to angiogenic switch, this is lymph angiogenesis and blood angiogenesis controlled by macrophages. So there are so many factors, and we worked on several of them. We also discovered, for example, YKI-49 is pro-angiogenic factor. There are so many factors which support angiogenesis and are produced by macrophages that questions a lot single VEGF-A, and we know that despite of potency of this factor, the therapy is, I would say, not such a potent when it comes to human application, because there are a lot of other factors which macrophages will produce, and sometimes if you block one factor, other factors are overexpressed to compensate for this deficiency. And macrophages also can build up a favorable matrix composition, degrading the matrix, degrading the structures under the vessels, that cancer cells can go out. And this and metastasize in the distant organs through blood. And here, there's also very interesting, I would say, observations, but not very well established technology to test it, that macrophages can build clusters with cancer cells, and these clusters will go to circulation. And this is how macrophages, together with cancer cells, will come together to metastatic niche and maybe gives instructions for the local macrophages to support this metastatic growth. So when we look for the number of protein factors here, you already see that making one single antibody, even kind of B-specific antibody, it's very difficult to eliminate the program. So the programs here have like a list of factors, which can, once again, compensate for the deficiency or blocking of a single factor from family. Yeah, so this is just one more deeper illustration in angiogenic factors. So we have three, I would say, new families, which are not deeply considered in terms of targeting of single members to stop angiogenesis. However, S100 proteins, semaphorins, and plexins, and also chitinase-like proteins have a strong potency to induce growth of new vessel in cancer. So this means, can you imagine how many antibodies you need to create to block at least 2-3 members from each family, in addition to VEGF, which we already know. So, and when we go to the programming of macrophages on signaling and epigenetic level, so of course, we all are experts in macrophages, we know the history. So it was subdivided due to in vitro modeling to M1 and M2 phenotypes, and then these phenotypes shifted to our understanding that this is vectors of polarization. And then we connected M1 to acute inflammatory in vivo phenotypes. However, if we think about M2, it seems like it's all the rest. So this means macrophages in allergy and tumors and in some chronic inflammation, they all will have kind of M2 signatures, because in all these healing and cancer environment, microenvironment, interleukin-4 and TBFbeta are frequently present. And these are, I would say, commonly accepted as major drivers of M2 vector of polarization. This is just to come also to the idea which was presented by the first lecture in our row, that actually these M2 macrophages, they keep these healing abilities because they are predisposed to heal. And these healing abilities should be considered by developing of the anti-tumor strategies. And here's one of example how it works, unfortunately, against cancer therapy efficiency. When we have chemotherapy, macrophages see chemical treatment as trauma. So this means, since all these agents are actually acting on dividing cells and macrophages are dividing in very limited way, they see chemistry as trauma, and they start to activate healing properties, which are more manifested, so I would say manifested in more specific way, depending on the chemistry is used. But what we know that, for example, doxorubicin will induce in macrophages to produce VEGF. So this means they try to heal the tissue, and they will start activation of angiogenesis. They can produce also cytokines which and microRNAs which will interfere with efficiency of chemotherapy effects on cancer cells, and will lead directly to chemo resistance in cancer cells. So, and this is also example of our study that we looked for the complete transcriptional programming in macrophages by cisplatin, and we found that it activates a complex, I would say, healing program and program towards tissue reconstruction, which remaining not killed cancer cells utilize in order to give a rise to even more aggressive cancer growth. And so this is like a background that macrophages should be targeted, but single factor targeting produced by macrophages is not very efficient. And this is why people tried, so a lot of researchers since 20 years, try 4 major approaches to target macrophages, not to allow them to support tumor. This first approach, historically the most, I would say, original, it's inhibition of differentiation and survival. Then it was inhibition of recruitment, then targeting of activation pathways, and using macrophage of the precursors as cell therapy tools, like a cell therapy-based approaches. So again, the primary approach to inhibit differentiation in survival was blocking CSF1, CSF receptor 1 axis. So everybody knows, so this is the most important factor which works during differentiation of pluripotent cells in bone marrow until the survival factor in tissue resident macrophages needed for the whole macrophage biology. And here I just made tables. We don't go in details, but I just want to show you how many clinical trials, alone or in combination with other things, were already running since last 20 years. And until now, the only approved approach is for tenosynovial Guyana cells in tumors. And also there's one approved which is running only in China for extrapancreatic neuroendocrine tumors. All the rest you see here, people tried it on all possible solid tumors, resulted in severe complications like fatigue, liver injury, a lot of intoxication, and actually the body does not tolerate the absence of macrophages, because you cannot, by targeting this pathway, you actually kill innate immunity in our body. So this means only in some very rare situations it can bring a benefit. Otherwise, it is rather toxic, and the toxicity is dominating over the therapeutic effect. So, and then the idea was that maybe we just inhibit recruitment of tumors, of macrophages in tumor, and this way they will not be able to accumulate and to support tumor growth. And again, there were a lot of attempts to target CCL2, CCR2, and also CCR5 and 6L12 and other receptors. However, it is now not coming to the clinical applications, and the phenomenon why it's very interesting. So if you block recruitment of monocytes to tumors with some antibody approaches, first what happens, for a couple of days it works, but then this system is so buffered by different chemotactic factors and different receptors that the body finds a possibility to overexpress other chemotactic factors or to make monocytes sensitive by increasing expression of other receptors. So, and then you have overwhelming wave of monocytes to be recruited in tumors, because tumors produce different chemotactic factors. So this means if you block one pathway, another pathway will compensate, but this compensation is much more stronger than initial ability of monocytes to migrate. So this means with this, we cannot also go systemically for blocking macrophage-mediated tumor support. And then it came to activation and functional pathways. And here, STAT signaling attracted a lot of attention. Also, TLR signaling attracted a lot of attention, and CD47 as immune checkpoint inhibitor was in clinical trials. And this CD47 mediated don't eat me signaling can be blocked also by antibodies. So it looked very promising to activate phagocytosis of cancer cells. So, and these are the clinical trials which reach up to phase 3 already. I would say it's the most promising what is going on. However, there are also complications, and there's also the mechanism which is very difficult to overcome here. And this mechanism is how to reach the specificity that cancer cells will be efficiently phagocytosed, but this will be restricted for cancer cell phagocytosis and will not lead to the phagocytosis of other healthy cells. So this means there are also a lot of complications here because macrophages become too aggressive. And this is also a very interesting approach here based on the targeting of CD40. And I think also, since it's only phase 2, this is can be done with combination with immune checkpoint inhibitors. I think this is also interesting schema how to bring macrophages to activate T cell immunity. However, I have concerns here, and I can share now my concerns with attendees of this lecture, that macrophage plasticity makes it difficult to link antigen presentation pathway to ultimate cytotoxicity, because even if antigen is recognized by T cells, if macrophages at the same time will produce tolerogenic signal, it can lead to the tolerance. And this is what concerns me all the time when I think about antigen presenting strategies. And if we cannot control macrophages the way that they are or silent, absolutely silent, or are very pro-inflammatory aggressive. And this is why I think that I would say understanding and controlling macrophage metabolism is necessary. I will come to our, I would say, initial trials to understand how it all works. After we will talk about using macrophages and their precursors as cell therapy tools. Here again, I believe that this is very promising approach, which actually these clinical trials are summarized here from 2018. But if you look historically, it was a great scientist in Regensburg, Andresen, who already got this idea in the early 90s, and they tried to program monocytes by interferon gamma in vitro, and then put it back in cancer patients and see if we can bring this anti-cancer potential because they are already programmed and they will migrate to tumor tissue, and then we start kill tumor cells. But now we all know, at this time it was not known, but now we know they are plastic. So this means whatever you do in vitro, so the most difficult part is how to fix the phenotype. How to achieve that monocytes, when they come in tumor, they are not instructed by the local factors. So, and this is our, I would say, general program for everybody who works on macrophages. So, in cancer therapy. But what we started from with progenitors and trying to program them to first by cytokines, but now there are attempts to make CAR macrophages. So CAR macrophages, I think this is really the most advanced approach. And you can also find antigens. You can find them in blood cancer. You can find them also in solid cancers. But I believe that we will meet the same problem that even recognition event does not guarantee the desired effector function if macrophages, even CAR macrophages, become reprogrammed. When they arrive the tumor site, when they actually should work. So, and this is just schematic presentation. So the CAR receptor should recognize, so it will be genetically installed, it should recognize, can be with lentiviruses, it can be with more advanced CRISPR technology integrated directly in genome, and then it will recognize antigens, and fortunately, this knowledge about antigen is accumulating. However, again, recognition doesn't mean necessarily the killing, and doesn't necessarily will mean phagocytosis if macrophages get the opposite instructions from tumor microenvironment. But here, I think if we manage to not even fix, but long-term control of macrophage polarization status, we can be very successful. So now, what are the major problems which people met by trying all these therapeutic approaches? When you inhibit survival, differentiation, recruitment, highly toxic or redundant pathways activated. When you target activation pathways, including toll-like receptor pathways, or low efficiency or plasticity interferes because of local microenvironmental signal. And using macrophages and all their precursors as cell therapy tools, low efficiency due plasticity or low specificity toward cancer cells. However, with CAR technology, we can overcome low specificity. Still, plasticity is a trouble. And now I would like to share, I would say, my thoughts and primary unpublished data regarding what is going on with macrophage metabolism. So, again, established knowledge, if we connect metabolism to the more classical understanding M1 of M2 dichotomy, we believe that in M1 macrophages, glucose is a major source of energy, while in M2 macrophages, we have lipids and fatty acids which are oxidized, and this is quick energy needed to kill bacteria. This is long energy which can be used for the slow homeostatic turnover in the tissue, maintaining the integrity of the tissue. So what we did during last 2 years, we tried to differentiate and make a modeling for different types of macrophages in culture. So we were first to demonstrate that interleukin-4 corticoids have induced separate programs. Interleukin-4 is inducing healing, corticoids are inducing a scavenging potential, and then we found that TGF-beta has a very complex signaling in macrophages because it's not only profibrotic, depending on the pathway activated, it can also be involved in metabolic control. So we found that TGF-beta can signal in macrophages through SMART2/3 and also through SMART1/5 pathways, and they lead to distinct transcriptional programming. And we already in 2005, we submitted the paper when we showed that if we take monocyte, differentiate to M1 macrophages, then give for 24 hours interleukin-4, the program is converting to M2 program. And if we differentiate for 6 days, M2 macrophages, and then on day 6, we give them interferon gamma or LPS or combination. And within 24 hours, you already see manifestation of M1 phenotype. And in this step, I would say it was already question raised, what is the background for this plasticity? And the idea comes since macrophages are here non-dividing, and you don't expect any genetic rearrangement like by T and B cells, and they are plastic. There should be some epigenetic control, which now gets a lot confirmation from various research groups that epigenetic is essential for macrophage programming. And when we talk about epigenetic, for macrophages, very important is not only DNA methylation, which is strongly dependent on the proliferation cascade, but histone code. Histone code is known for many years, and it has a lot of modifications which can be installed on different amino acids in histones. If you look for the number of amino acids, a number of modification, and then additional factor comes that it can be more than one modification on one amino acid. It can be also 2 or 3, and they can be heterogeneous. And these are relatively easy to install and also to remove. So the code is flexible. And this is probably what is the background for epigenetic control of macrophage plasticity. And here I summarize the study, which actually took us 2 graduate schools in Germany, like 18 years, when we found that hyperglycemia can install epigenetic code which is leading to pro-inflammatory programming on macrophages. So this all this study was done on human monocyte-derived macrophages in cell culture conditions. So when we started the study, we realized that actually everybody who works on macrophages in standard media will work in hyperglycemic environment because it has 17 millimolar glucose. If you go for ex vivo medium, SFM medium, standard medium, it is hyperglycemic. So we ordered glucose-free medium, SFM medium, and we supplemented it with 5 versus 25 millimolar glucose. And then we cultured macrophages under these conditions, and we looked for the transcriptome, and then we looked also for the histone code on the promoters of genes which were overexpressed in hyperglycemic conditions on macrophages. And what we found first, that hyperglycemia induces expression of CCR2, which is essential for monocyte recruitment in tumors. It also induces expression interleukin-1 beta, but also S100A9 and A12. And these factors are very important for endothelial inflammation, for endothelial permeability, and for angiogenesis. So this means by overexpression of these factors, monocytes have benefits by penetrating from the endothelial wall to the tumor, and also by inducing intratumoral angiogenesis. And then we focused here on these S100 proteins, and we looked if we have any epigenetic code installed by hyperglycemia, and it appeared that we have histone-free K4 methylation here, and also histone-free acetylation. We found also 2 enzymes which are involved in this histone code. And we also found, depending on the S100 protein, there is different duration of metabolic memory. So it was more pronounced in case of S100A12 as A9. And then we raised the question, okay, so if we go to diabetic conditions, and diabetic conditions predispose people to initiation and progression of different types of cancer, so we need to see how hyperglycemia cooperates with dyslipidemia. And here we came to the experimental content, which is not published yet, which I'm very glad to share with the audience, and to see if we find any possibility for the cooperation. So we decided to look for the cooperation of hyperglycemia and dyslipidemia, having in focus 2 major signaling pathways which controls macrophages not only in cancer but also in different inflammatory conditions, in atherosclerosis, in cardiovascular pathology. So we took toll-like receptors because they sense not only microbial products, which everybody knows that they sense LPS, while we can sense viral DNA and RNA, but they also sense triacyl and diacyl lipopeptide, which are the components of dyslipidemic profile in metabolically compromised patients. So, and we know that triacyl, triacetylated lipopeptides, which can be mimicked by PAM2-free synthetic components, and this is what we took in our model system, will drive TLR2-mediated signaling, and will come to the NF-κB activation, and will come to production of pro-inflammatory mediators. So toll-like receptors can drive inflammation once we see dyslipidemic ligands. Then we took our model system again, so I just presented how it works. So we took buffy codes, CD14 monocytes, and we put them in SFM glucose-free medium supplemented by 5 versus 25 millimolar glucose. And then after 6 days, we have these hyperglycemic macrophages, M0 and 1 and 2 prototype macrophages. And then we stimulate with dyslipidemic ligand or controlled LPS ligand, and then we can put TGF-beta 1 on the top and to see how toll-like receptors and TGF-beta signaling pathways cooperate. This is our model. So first, we go to the TLS themselves. When we did a lot of transcriptome analysis on the hyperglycemia, we have seen that even in M2 macrophages, despite that it's believed that M2 macrophages cannot utilize glucose, and this is, I would say, the tricky point we discovered, that even in M2 macrophages, we can see activation of transcription of some genes. However, it is very difficult to come to statistically significant data when you go for M2 macrophages. And this is, for me, actually understandable, because if you look for people with dyslipidemia in blood, or with increased lipid proteins in blood, or with increased cholesterol in blood, some of them develop atherosclerosis, some do not develop atherosclerosis. Some people have healthy obesity, even with high overweight, and some people have low overweight by already inflammation in the fat tissue. And I can imagine there is a lot of genetics behind, or maybe epigenetics behind. And this is what we see when we go for trying to identify statistically significant difference in transcriptome, the hypoglycemic conditions and dyslipidemic conditions. But here, what we found, depending on macrophage stimulation, if we put hypoglycemia in our culture, the different profiles of toll-like receptors are elevated. So in M0 macrophages is TLR1 and 8, in M1 macrophages 1, 2, and 6 are the most pronounced, but even in M2 macrophages, we saw TLR4 activation on the level of expression. This is not like 2, 3, or 5 times, but we are not talking about acute inflammation. We are talking of slowly developing low-grade inflammation. So this means even this, I would say, statistically significant, but full change is not so dramatic, can lead within years to the development of a pathology which predisposes our body to vascular complications or to the cancer initiation. And we also found that in hypoglycemic conditions, response to TLR4 receptors in M2 macrophages brings interleukin-1 beta on the higher level. And we also found, I'm sorry, this is, yeah. And we also found that if we look for interleukin-10, then in hypoglycemic conditions, this is not very strong decrease, but still statistically significant decrease in interleukin-10 production. And also, the same tendency goes when we take dyslipidemic ligand, not only LPS as a prototype ligand. So next, we showed that hyperglycemia amplifies TLR-mediated inflammatory response to dyslipidemic ligands. So this means if we combine hyperglycemia and with dyslipidemic ligands, then we have high level of toll-like receptor 4 expression itself, of interleukin-1 beta production, and also dramatic change in CCR2 expression. So this means hypoglycemia, dyslipidemia make macrophages more mobile. They can move more intensively in response to chemotactic factors. So this is a summary, actually, what we found. So we have a lot of toll-like receptors already on monocytes because they need to sense danger in the blood circulation. But when we put hyperglycemic conditions, we have elevated expression levels specific for each type of macrophages in macrophages after 6 days. And in terms of M2 macrophages, it leads to elevated addition of dyslipidemic ligand, elevates TLR4, elevates interleukin-1 beta, CCR2, and suppresses interleukin-10. And then we decided to look how hyperglycemia and dyslipidemia affect TGF-beta signaling. And what we first found that hyperglycemia delays SMART2 free activation in M2 macrophages. This is normal glycemia and hyperglycemia. When we go for dyslipidemia, then it blocks TGF-beta SMART1/5 phosphorylation. So this means we have here hyperglycemia works on one arm of TGF-beta signaling, dyslipidemia works on another arm of TGF-beta signaling. Then we showed that TLR1/2 inhibition restores SMART1/5 phosphorylation. So this means at least so when we go for dyslipidemia in mediated suppression of TGF-beta SMART1/5 pathway, TLR1/2 is involved, and this is the sensor for dyslipidemic ligand. When we showed that PAM-free dyslipidemic ligand increases MYD88 protein stability, and this was the first idea that something can go on ubiquitin system level. When we looked for the complete profiling, or KEC-based profiling, so this is RNA sequencing, and we found that if we go for normal glycemic conditions, dyslipidemia plus TGF-beta have activation of a lot of inflammatory pathways, and it's even more pronounced when you add hypoglycemic condition. But dyslipidemia alone is pro-inflammatory. With hyperglycemia, it is much more dramatically pro-inflammatory. This is just a selection of some genes, which we showed the transcriptomic signature of TGF-beta signaling macrophages under diabetic conditions. And again, when we go for TGF-beta signaling, major effects is provided by dyslipidemia. It has a lot of pro-inflammatory effects. Hyperglycemia will amplify it. So dyslipidemia is like a primary here. And then this is, I would say, work in progress that we are now. So we took from all this RNA-seq data, all what is related to ubiquitin pathway regulating genes. And we see that we have normal glycemic conditions. When we put dyslipidemia on, it provides a lot of it provides the changes in ubiquitin system. But if we add TGF-beta to the system, the number of changes is of another magnitude. So this means TGF-beta is a very strong regulator of ubiquitin system in dyslipidemic conditions, but under normal glycemia. If we switch to hyperglycemia, this picture does not change much without TGF-beta. It's very similar. But when we go for hyperglycemic conditions plus TGF-beta, TGF-beta cannot change the profile of ubiquitin system regulated genes. The changes are much less pronounced if you compare this picture, the number of regulated genes, and this picture. And this is the point we are now. And of course, we would like to understand how this cooperation of toll-like receptor and TGF-beta pathway in the real dyslipidemic hyperglycemic conditions, which are typical for people with obesity, unhealthy obesity, metabolic syndrome, and type 2 diabetes. What we can extract from here, from this regulatory pathway, to find the targets from ubiquitin system, which can be used in order to control macrophage phenotype in the desired way, do not allow macrophage plasticity. So, and this is what I wanted to share with you. We came to the level of regulations. I don't offer now any clear targets, but we are ready to, if you have model system and you would like to address how ubiquitous system can be targeted, so we can go together through our data and see which pathways can be validated in the patient's material or in the appropriate animal models. So this is what schematically how we see now how all this works. So we have toll-like receptors. So pink is hyperglycemia, yellow is dyslipidemia, and these are the contribution to the whole system in macrophage. So under hyperglycemic conditions, you have more telomeres. When dyslipidemic ligands come, they drive MYD88. However, dyslipidemia does not allow it to be degraded in proteins. So, and this is only one manifestation of a broken ubiquitin system. So you have more MYD88, it cannot be degraded. Proteasome, pro-inflammatory pathways, despite of a small difference in telomere profiles here, we are leaky. Then dyslipidemia also blocks SMAD1/5 pathway, and hyperglycemia blocks SMAD2/3 pathways induced by TBFbeta, and then altogether it also blocks proteasomal work. And then we have leaky pro-inflammatory pathway, and TBFbeta cannot interfere with this inflammatory pathways anymore. And now I shortly present one more story about another metabolic regulator, because I would say people believe that M1 macrophages are always anti-tumoral. So this means if you induce inflammation, it will work against tumor. And I would like to present you the data which are actually against this from the metabolic point of view. Because M1 macrophages use glycolysis. So logical next step would be, if you have more glycolysis, you have more inflammation, it will work against cancer cells. However, we have a lot of indications now from the statistical studies, and I believe the researchers involved in discussion can know even more about this than me, that obesity, metabolic syndrome, and type 2 diabetes programmed by cytokines, by lipid mediators, program already circulated monocytes to be predisposed to support tumor growth. They bring some programs which do not allow them to fight against tumor, but they provide not silence, they provide chronic inflammation. And what we did, we wanted to know if there are any mediators, metabolic mediators, which potentially can first be biomarkers that this metabolic indicators a biomarker or that monocytes are predisposed to fight against tumor or to support tumor. And second, maybe they can be also serve us as a target. So what we did, we compared colon and rectal cancer monocytes and also tissue macrophages. So we did it for flow cytometry, then we did RNA sequencing and validated by real-time PCR. And what we found here, there's one very interesting mediated, which is named PFKFB3, which is elevated in monocytes of patients with colon but not rectal cancer. And this is pro-glycolytic factor. Without this factor, glycolysis is not efficiently running. So actually, you would expect that this factor should indicate the ability of immunity to fight against cancer because it allows to use glucose and to do inflammation. And then we went to the correlation with clinical parameters, and then we also made spatial transcriptomics by nanostring technology. We found that PFKFB3 correlates with bad prognosis, with bad therapy response. So this means low-grade glycolysis which monocytes bring already in form of overexpressed pro-glycolytic regulator, actually supports pro-tumoral activity of monocyte-derived tumor-associated macrophages. And then this is how it looks like. So you have we have proven here with PFK positive monocytes, because this is staining for CD14 and this is for CD68 in colon tumor tissues, where preferentially infiltrating the tumor site. And these are, and these are the clinical data. So if you have KFP3 expression in tumor tissue, so in macrophages, because we did spatial transcriptomics, so we were able to select for our analysis PFK3 expressing monocytes and macrophages. So you have more of recurrence of colorectal cancer, you have higher tumor size, and overall survival goes down. And then again, point for discussion. So you see the factor which is supporting bad, probably low-grade glycolysis in tumor-associated macrophages can be targeted. So this is more like a screening study performed by mass spectrometry. So we used inhibitors for PFK3, put these inhibitors on our monocyte-derived macrophages, and then we made a lipid profile to see if lipid profile will change to the beneficial direction. But unfortunately, it was not. So this means we have here inhibition of PFK3 leads to the increase of sphingolipids, linoleic acid metabolism, and also arachidonic acid metabolism, which probably will work in pro-tumoral way. And here again, this is work in progress. We try to understand what is going on. We are not coming with ready targets here, but I would like to point out that targeting of single factor in one single pathway might not be the way to fix macrophage metabolic phenotype in the desired direction. So I think we can think about the major signaling pathways which are controlled by metabolism, and we can think about glycolysis. But in terms of glycolysis, or it should be very shortly activated on such a top level that macrophage kill everything around, but this is acute action, or maybe it should be completely removed from macrophages, that they are not able to support tumor growth just because they don't have any energy for this. And these are the points which I would like to discuss with you. If you find arguments towards this hypothesis, or you see it in a different way. How I would finish the lecture, that 6 days do not allow you to look for the memory and also the programming of metabolism, and also for connections between metabolism and epigenetics. This is why we develop a new model. In this model, we can keep macrophages from monocytes, develop from monocytes for 36 days. And we analyzed already on all these days that they keep major activity of macrophages to be responsive to LPS. We keep macrophage biomarkers, so they are all CD68 positive here. We made RNA sequencing comparing day 6 and day 36. We see now that there are strong epigenetic changes happening during this period, but if we think about resident macrophages, they are not 6 days macrophages. They are much long-term differentiated macrophages. So, and what I believe that this model system will allow us to address the questions and to test different types of modulation of macrophage metabolism, then analyzing how this modulation of macrophage metabolism can affect epigenetic memory, and if we can, by modulating metabolism, can we fix, at least for the middle term period, macrophage phenotype, and then we will know how to instruct CAR macrophages to do the job properly without being inactivated by a local tumor microenvironment. So this is now, I would say, our working hypothesis, how we should go on. I would like to thank number of people. This is not all people, but number of people who contributed to this macrophage metabolic story about epigenetic control by hyperglycaemia in macrophages, and also our funding bodies and long-term cooperation partners from Groningen, who were partners in the diabetic macrovascular complications consortium, and who studied effects of hyperglycemia from different sides, from endothelial cells, but we joined our forces in modeling also macrophages in metabolic conditions. And also I would like to thank German Red Cross Blood Supply Service, because this is a great organization which is providing us with internal grants, very supportive in the difficult time. We all experience, I think, the funding, and so we can continuously support our mainstream idea about controlling macrophage metabolism and looking for new and new targets. So I would like to thank you for listening, and I will be very glad to hear critical questions and critical comments to our understanding how we can go on with macrophage therapy approach. So thank you very much.